So coming after Alex is a tough act to follow. But the reason that I gave myself this second talk was to keep company to his talk is after I saw Alex's abstract. And in particular, the first part. So I want to refocus our attention on the first part of his talk where he talked about information, the information-seeking task that came from animal behavior. And recently in the monkey field, there is a resurgence of the use of this paradigm in various forms. Some of you may know about one study by Ethan Bromberg-Martin and Hikusaka, who did something very similar with monkeys, a very similar experiment. I won't talk about that experiment today, but I'll talk about some variations of it and some work that we have done that go in the same direction. And the underlying logic is to exploit this useless information paradigm, seeking useless information. As I've told you before, looking at when we study information seeking in the lab, we were faced with this problem that in most natural behaviors, animals use the information to increase their rewards. And so if you want to understand it, is there something special? Do we have an intrinsic desire for information? You somehow have to pull these apart. And all right, so just to remind you, in yesterday's talk, I considered an active situation such as this. You have uncertainty, you need information, right? So you have to make a decision, you have a moment of deliberation, you sample information, you gather, let's say, visual information, just for an example, you incorporate that, and eventually you make an action decision, right? This was the scenario. The action has rewards and uncertainties related to it. And the question is, how do those rewards and uncertainties feedback and influence this information sampling step? All right. So we've seen, I think yesterday we've seen two talks, two ways to possibly dissociate those. I talked to dissociate information gains from reward gains. I talked about one way. Michael Frank talked about another way when you seek information about the state of the world, which, but you've seen that in both cases it's difficult. You have to go through some experimental contraptions and model the data and so on and so forth. It's non-trivial. So another approach that people are taking is to say, okay, forget the action, right? Let's have a more passive context in which you don't really have a goal. You're not trying to achieve anything. You're just kind of thinking about stuff, right? Okay, so you can see clearly the problems with this, right? I mean, if you put people in a lab and say, just look around, well, it's not, right? It's not exact. You get all kinds of strange behaviors. And moreover, it's worse than that. It's not that people aren't doing anything. It's just that you don't know what they're doing in their head. Okay, so then the approach that people took. Okay, so here's the question. All right. So this is the question. Maybe I should go through this quickly. I'm sorry. I got confused in my slides. Okay, so what happens when, so just to go reintroduce LIP really quickly. So again, yesterday I told you that LIP selects targets for goal-directed actions. When you have a goal-directed saccade that serves a certain action, you have this top-down mode of selection. But in addition, we know that the same neurons also respond to a variety of factors that don't seem task-related, such as salience. The sudden onset of something, even if the monkey knows it's a distractor and it can only interfere with his task, that will be registered by the cells. There's been a social information task, affectionately known as monkey porn, where monkeys traded juice for pictures of behinds of female monkeys. And the other factors, the three factors that I want to talk about today is again reward and novelty and uncertainty. And I will ask, how do these factors impact salience in a context in which the stimuli are associated with these properties, but they don't really allow animals to act on them so as to increase the reward, right? Okay, so schematically here, I look at reward, and so now instead of talking about the operant reward of an action, I'm going to talk about stimulus-reward association. So if you want to call them Pavlovian values, right? Pavlovian values, and I'm going to talk about novelty, like something that's, I mean, that's a picture of strange sheep. That's my signal for novelty there. Right, and the first questions I'm going to ask is, do LIP encode value-driven attention capture? So this is a phenomenon that we have more and more evidence from human psychophysical studies, which say that by training stimuli, by conditioning them with rewards, those stimuli acquire intrinsic salience, and they will be more distracting even when they're irrelevant to a task. Okay? And then I will ask, so I'll talk about Pavlovian reward and novelty in the first part, and then I will ask, well, how do these really interact with uncertainty reduction? Because they really are two different categories of things. Okay, so the general strategy will be, right, so a first problem you have is, okay, you want, you're in an sort of non-operant context, and we need to grab our subject's attention with some stimuli in a manipulable way. And then, you know, it's really hard to know what our subjects are really interested in, right? It's hard to know for people. It's also hard to know for monkeys. So the one thing that we can be sure that they're interested in is actually, that's why we go back to rewards, is knowing what reward will be coming. So again, we're in this paradigm where we use reward, and what we do is we have the monkey start off in a lottery situation. So every trial, let's say they have a 50% chance that they will get a reward or not when they start the trial, right? So at the end of the trial, they get a reward or no reward. And there's nothing they can do. In this sense, it's a lottery. The computer selected it, and that's the end of it. But in the middle of this waiting time, we allow them to sample a cue, right? And that cue can provide them advanced information about what will come. Right, so it's the early resolution of uncertainty, a lot like what Alex spoke about. Okay, so let me go now through one paradigm, one way in which we use this logic. It was the following. So what you see is the screen that the monkey saw from top to bottom. The monkey began by achieving fixation up in that top screen, and we were looking at an LIP neuron with a receptive field at the dot. And then the monkey saw the reward cue, right? And the reward cue could flash either in the receptive field or could flash at the opposite location. The location was randomized, and its shape was also randomized. And we used cues that we had pre-trained to indicate. So some cues, I will mark them in blue, they were positive in that they predicted that a reward will come. They were good news. Other cues that I'll show in red were bad news and predicted that there will be no reward at the end of the trial. Okay, so now notice that we put the stimuli in the visual periphery, and our goal was to see, do they bias attention, right? There's no reason in our test, there's no reason for the monkey to actually orient towards the stimulus. He's not rewarded for it, but it could be that the stimuli automatically bias attention. Okay, so now, so we put them in the periphery, they might bias attention. Now we need to measure whether they bias attention. And here's how we did this. After the stimulus was extinguished, there was a delay period. The monkey just had to fixate in the dark for a minute, for half a second or so. And then we showed him, okay, sorry, that reward stimulus told him what will happen at the end of the trial. Okay, so then the monkey had to wait. And then a second target came on. And the target could be either at the same location as the stimulus or at the opposite location, randomly. Okay? And the monkey had to make an eye movement to the target as quickly as possible. Right? As quickly and accurately as he could. Okay, so notice, remember that the location of the reward stimulus was randomized, right and left. So the location of the target was also randomized independently, right and left. So the stimulus location told the monkey nothing about what he would have to do. A perfect actor in this case would simply make an eye movement to the target as quickly as possible. That's the optimal strategy in this task, right? On the other hand, if the attention is biased by those reward cues automatically, we can detect that, because let's say that a cue attracts attention to its location, then the saccades made towards the location of the target would be facilitated, a saccade made away would be suppressed, and the other way around. So that's our reasoning. The saccades are our metric of whether the reward cues biased attention towards or away from their location. Okay, now another question you may have here is, let's say you have an unrewarded trial, you saw a negative, a CS minus, you know that there's no reward coming, why would the monkey complete the trial? And the reason is they'd rather not complete it, and indeed they try to break fixation, but we immediately repeat the same trial until it's correctly completed. So you can think of the reason to complete a non-rewarded trial is to proceed to the next one. Okay. All right, so what we see is that LIP neurons encode the reward-based salience of those stimuli that predict the rewards. So here are the responses to the onset of the cue, right? The black bar is the 300 millisecond period when the cue is on. After that, there's a delay period when the monkey is simply waiting to be told where to make his eye movement. And you can see that the neurons have, for both cues, they have a fast visual response, and then they also maintain the sustained activity during the delay period, right? Remember, even though that stimulus was irrelevant. And when you examine that sustained activity, it turns out that it is specific for the location of the stimulus, right? So if you look at the positive stimuli, and now the black trace in that inset is the same as the blue trace here, and the gray trace shows what happened if we put the cue outside of the receptor field, right? If the cue appeared outside of the receptor field location actually had a little bit of a dip. And if the cue was in the receptor field, those neurons maintained a sustained excitation, right? So this is not global arousal or excitement when you expect a reward. This is an actual spatial bias towards the location of that positive stimulus. And now if you look at the negative, this is the more interesting of the two. What you see is that when the cue appears in the receptor field, there's a visual response, but then there's sustained suppression at that location, right? So this is a sort of negative salience. And then if the cue appears at the opposite location, you see that the activity stays pretty much the same. So again, it's not a general decline in motivation. Oh, I feel bad that I don't get a reward. It's an inhibition of that visual field location. Okay, bad news, inhibit attention at a particular location. And we can see this in the eye movements that the monkeys make. Remember, the eye movements are our diagnostic of these attentional biases. Okay, so what I'm showing you here are every gray dot is the end of a saccade that our monkey made in two different conditions. The target is at 1-0. And you can see these are very highly trained monkeys, and these are very easy saccades. The target is bright, visible, it stays there the entire time. So what you see on the top is how the monkey performed on a trial in which there was a positive stimulus appearing at that location. You can see that's the level of performance that we expect. And on the bottom, the saccade metrics and everything is the same, except that there was a negative stimulus appearing at that location half a second before. So it really is striking how poor our monkey is. So that, by the way, is the location that shows the inhibition in the parietal cortex. Notice that often the monkey is trying to look away, directly opposite, but also many times he's trying to look towards the cue, and somehow he seems to be dysmetric. He just cannot put his eye on that location, right? And this is just the average data. If you look at saccade accuracy, If it's a positive, a good news stimulus, performance is good. Whether the saccades are made toward the cue or away, it's a little better if you move towards the cue. That's the attentional attraction. If it's a bad news, performance is okay if you ask the monkey to look away, right? Even though these are unrewarded trials, he does okay as long as I can look away from that stimulus. But if you ask him to look towards that location, he's really impaired. And we see the corresponding things in reaction time. Okay, so it seems that, now remember that these error saccades cost the monkey real juice, because they are scored as errors, which means he gets another unrewarded trials, and he reduces his total rate of reward. So this is really irrational behavior. And yet, and as I'll show you in a minute, it increases. The more you train the monkey with these stimuli, the more this irrationality increases. Okay, all right, so this brings me to the second part of this experiment. Everything I told you so far was based on highly overtrained cues, right? So those are novel shapes, abstract shapes that the monkeys hadn't seen, but we trained them for tens of thousands of trials. Some predicted reward, some predicted no reward. So very overlearned stimuli. Together with the stimuli, with those overlearned stimuli, we introduced new shapes in every session. And we presented each, and we assigned some shapes to predict positive news, some shapes to predict bad news. And we presented each one for about 30 times to allow the monkeys to learn their reward associations. So now I'm going to tell you about the responses to those novel stimuli. Okay, so what we found when we looked at those novel stimuli, first of all, we monitored, in order to make sure that the monkeys understood the reward prediction, we monitored their anticipatory licking, right? So in other words, there was a delay period between the time when the monkey saw the cue and knew what was coming. There was a delay period, and eventually the trial ended. And monkeys often anticipate. If they know that if they expect a reward, they will start licking ahead of time. If they expect no reward, they will really withhold their licking. And what you see here is measurements of that licking as a function of the number of times that the monkey saw a particular cue during a session. So first, if you look at the pale curves, those are for the familiar stimuli, positive in blue and negative in pink. You can see that clearly the licking distinguishes between them from the first presentation. Of course, the monkey knows what they are. The novel stimuli are in the darker colors, and you can see that in trial 1, the monkey optimistically licks. But then, if a stimulus turns out to be bad news, the licking extinguishes very quickly, within 3 or 4 or 5 presentations. So the monkey very quickly learns what is good news, what is bad news. And the surprising result was that the visual response, so you would think that the visual response would also show that suppression just as quickly as the monkey learns, but that wasn't the case. So in fact, the visual response remained high for a longer period of time. So you can see that there, if you look at the unrewarded cues, the response to the familiar ones is in pink now, and you can see the suppression. But the response to the novel cues remains elevated and doesn't show the suppression. In fact, it's a little more similar to the positive cues than it is to the negative cues. In other words, the salience of a novel pattern remains high for a while, even after the monkey learns that that pattern is unrewarded. So here's a quantitation of this. If you do the same plot that I've shown you before, except now you plot the visual response, and you can do the same with the delay period activity, you can see that the novel minus response, maybe it has a little bit of a decline, but it stays closer to the positive cues, and it doesn't go down to the level of the highly overtrained. So in other words, after a while, so we expect that if you train a stimulus as predicting bad news, eventually it would acquire this negative salience. But that takes a relatively long time to occur. We didn't measure the time that it takes, but we estimate, based from other evidence, that it's around 200 trials or so. Okay, and we can see this also in saccades. So On the top is what I showed, an example that I show you, very poor saccade behavior following a familiar negative cue. Following a novel negative cue, though, the saccades are better. Okay. All right, so what do we learn from this? So we think it's something like this. If you have a stimulus and you start from novel to familiar, and let's say it turns out to be a bad news stimulus, you initialize it high. So if you talk about motivation or decision making, initially you're motivated to approach that stimulus, and as you quickly learn that it is bad news, and then you extinguish that approach. So, for example, licking behavior or motivated behavior. And for salience, a similar, it also extinguishes, but a little bit slower, right? So if the salience of a novel negative stimulus is relatively high, after a lot of training, it becomes negative, right? It becomes suppressed. So, which suggests here that we have some sort of window when things, even if they are unrewarded, there's still salience, and maybe it's just a more protected window for a little bit, to give you a little more time to learn about this stimuli. Okay, so going back to our scheme then, so what we've seen is that we have these, even when an operant reward is not involved, stimulus reward associations can increase salience, and also novelty can increase salience independently of reward. Okay, so now, right, let's look at the third question. What does this have to do with uncertainty, right, and information seeking? So, you know, the salience effects are very puzzling, because as I've shown you, they occur even when they're not useful for a task, and they're very persistent, and they don't extinguish with training. In our case, the reward effect became stronger with training rather. So this suggested to us that it's a very sort of fundamental and important mechanism. And the one way to understand it is that, well, you know, we do need to engage with sensory stimuli in our world in order to learn about our world. And so the natural next question is, well, are we really trying to obtain information in the strict sense of reducing uncertainty, right? Okay, so in order to do this, we're going to a little bit of a different paradigm. We're still in a lottery situation where the monkey starts with some uncertainty, and at the end of the trial he get a reward or no reward. And in the middle, we let him get a sensory cue about what will happen. One difference is that now we're going to give the monkey a search display and ask him to work a little bit to find the cue, so we can get a measure of how motivated he is to actually find that information. So that's one difference. A second difference is that before we used the fixed probability of 50% probability of getting a reward. Now we're going to vary the prior probability a little bit, 0, 50, or 100%, in order to manipulate how uncertain the monkey is about his reward. Okay, so here's how this okay, maybe I'll come back. Let me come back. Let me show you the task first. So here's how the task goes. The monkey begins by fixating, and then he sees a first cue that tells him the prior probability. And those cues are, again, pre-trained, and they can signal 100%, 50%, or 0% reward. And again, the cue signals the reward, and that's it. There's nothing the monkey can do about it. In between, while he's waiting, he gets a search display. And it's a display of placeholders, and his gaze is free to do as he wants. And the rules of the game are such that if he looks at one placeholder and holds his eye there for a minimum fraction of time, that placeholder turns and reveals what's underneath it. And two of these have just a meaningless pattern, but one of the placeholders contains a second cue, right? And that cue gives the monkey deterministic information of whether it is 100% or 0% reward. So that cue can fully resolve the uncertainty. The monkey doesn't know where it is, so he has to search, right? And we give him time. In principle, he can look at all three placeholders if he wants to. Okay, so, all right, so here's what the monkeys are doing. So this is the search function. So what I'm plotting here is the percentage of trials in which the monkey actually found the second cue that was hidden there. And on the bottom is the prior reward probability. And you can see that it's this sort of skewed, inverted U-shaped function. In other words, the monkey searched the most when he had the highest uncertainty at 50%. And he didn't search at all if he knew ahead of time that there was no reward. He didn't even look at the search display. The most surprising thing is that the monkey also searched very vigorously when he knew for sure that he had a 100% probability of reward, right? So the 100% case, there's zero uncertainty, there's no reason he should be looking for a second signal, and yet he really wants it. This is monkey 1. Monkey 2 showed a similar pattern. He was, as I'll show you in a second, that monkey searched a little less overall, but when he did search, he had a similar pattern. So what this suggests to us is a combination of influences of reward and uncertainty, right? So if you look on this axis, if you plot on the x-axis probability, if you look, expected reward is a monotonic function of probability. Uncertainty is an inverted U-shaped function. So if you add these two, you get sort of the skewed functions that we that we get. So it seems that both reward and uncertainty motivate visual search, and they probably can have different weights on different days, and so on. Okay, another question that this allowed us to examine is the monkey were absolutely free to search or not to search. So if they wanted to disengage from the task, that was fine too, they would get the same rewards. And we have two monkeys, they did different things, right? So monkey 1 was our star performer. So you can see that, so these are the three conditions. The dotted red is 0%. Now the x-axis is the number of testing days. At 0%, he didn't search. At 50%, which is the solid red curve, he searched. At 100%, I'm sorry, 50% is the blue curve. He searched at 50% is blue, 100% is red. So he's searching, and he continues. He actually increases with time. He really wants to find that signal. Monkey 2 started off by doing this task not very enthusiastically, and when he did it, he had the same order, relative order of the curve. But gradually, at the end, he completely stopped. So he figured out that he doesn't need to search in order to get his reward, right? So we talk a lot about intrinsic motivation to obtain a signal or obtain information. Well, it seems that this varies by subject. This varies by subject, and this shows you also the pitfalls of looking for this in the lab, because we're all used in order to record our neurons and so on. We need reproducible, reliable behaviors. And we'll all shape our behaviors very strongly with rewards. But if we give up that lever, then we have to get around problems like these. So we're still thinking what to do with our monkeys. What to do with that monkey too, who's too smart for us. Okay, and the last bit I want to tell you, so we saw this funny U-shaped function. And we have some preliminary evidence that LIP neurons encode the same pattern. So here are the responses to cues that predict different probabilities of reward. So you can see the red and the blue curves are similar to what I showed you before, and they show the 0% response to a bad news cue and response to a good news cue. And the surprising fact is that if you have a cue that is uncertain, the 50% cue, that produces stronger responses. So in the neurons, we also have this skewed inverted U function. All right, so what are we left with? Okay, so I want to just say a word about this sort of Pavlovian bias to positive stimuli that was really surprising in our data. And because again, it comes up also, there's psychophysical evidence for it in the human literature. In many different tasks, it seems that people are automatically captured by stimuli that have prior reward associations. And often this is portrayed as, of course, when we are in the world, in big environments, we want to orient to things that are good or potentially good. But I just want to emphasize that simple good association is different from information. Right, so here you have, I thought this cartoon was made just for this purpose. Here you have a very strong Pavlovian stimulus. Maybe it's good in general to track that sort of thing, but not always, right? So this person would have been better served to look ahead. And I like this wise man once said, that desire is no substitute for knowledge and enthusiasm is no substitute for information. So just to make that distinction. Okay, so right, so when we talk about information seeking in looking behavior, I'll put a star next to it because it's not always information that we seek in the sense of uncertainty reduction, or at least, right, we just sometimes just like to look at things for the hell of it. Okay, so what have we learned about this open-ended visual sampling? It is guided by heuristics that can be determined by previously learned stimulus-reward associations and novelty. Also involves a desire to reduce uncertainty. These are separate things. And individuals vary in their intrinsic motivation to actually sample signals from the world. And that's it. Thank you.
Questions?
Can I ask you? So the monkey one behaves more or less as you would expect from an auto-shaping Pavlovian analysis. So it's responding to the cue in a Pavlovian contingency, that is, you don't need to do anything, but you address behavior to the signal that actually announces it. Monkey 2 is kind of more operant. So can you really, I understand the logistics of working with primates, but can you really tackle variation of what makes an animal respond to Pavlovian contingencies or not with two animals? So there is a problem here. You ought to ask for
we need more animals.
Money or whatever.
That's true. That's true.
8 animals.
That's true.
Something to make, because I don't know what to make of the fact that 2 animals have this very different response.
Right. Right. Exactly. I feel the same way. I mean, for us, it is an interesting question, and we need another approach to address it. Right. I mean, yes. So that is one of the things on my list that I may want to do at some point. Yeah. Yeah. Yeah.
I have one question in the first experiment. So you said the probability of receiving a reward was 50-50. But in order to get the monkey engaged, in case he bailed out in that trial, you would repeat the trial. So in the next trial, the probability of receiving a reward is 0.
Only if he made an error. If he completed the trial correctly.
So, but then overall, he will have, because the error
it's less, yeah.
Punishing, so how do you kind of account for that?
It's less. So it's less. I mean, we did various analyses looking at some days monkey performed better or worse. The daily variations were minimal. So overall, what would happen, I guess what happened overall, his overall probability of reward was less than 50-50, was 45 or something like that. But as long as it was constant, you know, we could work within that. And we did various controls to look at the number of unrewarded trials that preceded a final successful trial and so on, and we didn't find much influence there. Yeah. Yeah. So Alex, so I found it very interesting in your talk, the fact that even that when you signal the good news without a stimulus,
Yeah.
That was quite interesting. And two things were interesting. First of all, the animals did go for it, but they went for it much later.
Yeah.
Than when there was an actual stimulus there.
Yes, the striking.
So I wonder, right?
For me, the striking outcome of that experiment, and I remember mentioning this with other people at coffee, is that when the animal at the beginning doesn't know very much, it's choosing optimally, responding to the two probabilities.
Right.
But as it learns,
right,
then it flips completely to a very irrational behavior. But in our case, it's costly.
Right.
In your case, it's not clear what the cost is of this responding.
Right. That's right. That's right. So that's why now, right? We will. That's what we're doing now. Actual trade-off tasks of information and no information. Right. Right. So. Right. Okay. We'll talk more in the break. Thank you.
We still have 1 or 2 minutes.
Oh, okay.
I would like to ask one question.
Yeah. Okay.
What's your interpretation of why monkeys like good news?
Why monkeys like good news?
Yes, what is your hypothesis about? Like, I think like Alex noted one hypothesis of why the birds in this case they like good news. I mean, do you have? I mean, do you share this view, or do you have some other idea? Or?
Yeah, no, I do share this view. I think that being biased toward positive stimuli is a useful strategy in general, right? And in an operant context, we are biased to stimuli that increase our future reward, right? That's the definition of an informative stimulus, and that's the reason to look for information. So I think that it is actually built in as an easy computation to make. Also, this Pavlovian relationship is a computationally easy one to make. To estimate when you have uncertainty, when you reduce uncertainty, I think that's a much more laborious cognitive computation. So I think we actually have both mechanisms that are playing there. Sometimes the Pavlovian one, it's maybe even stronger because it's easier.
Mm-hmm.
So yeah.
I'm reminded of the omission schedules used by Williams and Williams, if you remember those. So these are cases where an animal has a signal, and if it doesn't respond, it gets food. If it responds, it doesn't get food. So it's very straightforward. All it has to do is do nothing.
Right.
And in close to 50% of the cases, the animal responds and loses the food.
Right.
And that's because the strength of Pavlovian responding is so strong.
Yeah.
That they are shaped to normally address behavior to something that is anticipating that. And this is self-regulatory because obviously the more you don't respond, the more you experience the contingency.
Right.
And I think your monkeys are facing something similar to that. It can be coached in those terms, and it would be worth analyzing. But what to do when two individuals do completely different things is I don't know how to handle that.
Right. Okay.
Okay, thank you.
All right, thanks.